Today's speaker, Dr. Fatima Mroue, I'm lecturer in the mathematics department at the UB. She got her PhD in applied mathematics, working on problems to do with electrocardiophysiology, particularly looking at the electric mechanical coupling in heart physiology. She's worked on developing the models, studying solutions, developing algorithms, studying the stability of algorithms, the whole works. So certainly have an expert on the field with us. She applied to the CAMS fellowship, and particularly on a project associated with emerging interests in bacterial colonies and their growth. This interest is new to her. When we asked her about this change in direction, she was very clear. It's a context in which experimental data is readily available, and she's very keen on putting mathematics in confrontation with reality. One would think that with all the interest in heart physiology in Lebanon, data would be available, but apparently this is not the case. We're very happy to have her with us. Fatima, as part of her application for the fellowship, has proposed to structure a whole workshop dedicated to mathematical biology, which will unfold in the fall of 2022. I just heard that there is significant progress on this project, and we're very much looking forward to it. I want to thank in this context the collaboration with Math Chair Professor Florian Bertrand, who was very supportive of Fatima's project and the time commitment that it requires. So Fatima, without further ado, the floor is yours. Thank you very much.
Thank you for the introduction. Hello, everyone, and thank you for showing up today. So the topic is about mathematical modeling of self-organizing bacterial communities. So it's a topic clearly in mathematical biology. I will start first with a brief introduction about mathematical biology in its historical context, but the introduction is not just for the goal of the history. I have another goal in mind, which is to lay a conceptual framework that motivates the modeling strategy that we have in mind for the bacterial colonies, for the organization of the bacterial colonies. So you all know that in the 20th century, physics was nurtured a lot by the use of sophisticated mathematical tools, and the cross-fertilization between quantum mechanics and functional analysis led to the term mathematical physics. Well, at almost the same time, there was an attempt to start mathematical biology as a discipline, and it is widely accredited to Darcy Thompson, who was a biologist and at the same time a mathematician. This intuition of structuring mathematical biology just as mathematical physics is structured. So Darcy Thompson, in his book on growth and form, wanted to introduce the notion of the effect of physical parameters and chemical parameters on the generation of form. So for example, here in his book, he shows that by simply applying a 20 degrees shear mapping, one can change from one species of fish to another species. And this is really related somehow to his critique of the Darwinian theory of evolution. And the way how he described the mathematical beauty of forms that originate in nature, like in plants and animals, really was or stimulated the interest later, or the scientists like Alan Turing, among many other scientists. So this is from one hand, from a biologist perspective, also from a physicist perspective. In his book, Schrödinger, which is called What is Life, Schrödinger looked at also the genetics from the point of view of physics. So he said that there must be the existence of an aperiodic crystal inside the cells in which the genetics is stored in the chemical covalent bonds. And this book was accredited also by later Watson and Crick, who discovered the double helical structure of the DNA as the source of inspiration. And in this sentence, living systems have the ability to extract entropy to keep their own at low levels. Schrödinger clearly affirms that there is something special about living systems. They have their own abilities. They have their own framework, if you want. Along the same lines, the Nobel laureate Lee Hartwell in physiology and medicine clearly affirms that whenever we want to use mathematical structures in the case of biology, one cannot directly use the same structures that apply for the inert matter. So, there must be some kind of mathematical structure that takes into consideration the particularity of the living system. So although we have so far for that long period of time till now, we have seen many contributions of mathematics and computing in biology and medicine, from whether it is from the computing point of view or from the theoretical point of view, the mathematician working in biology is still facing many challenges. The first one is the absence of a field theory. So in physics, there is a field theory that guides the mathematical approach. In the case of biological systems, this is still not the case. And as a result, one needs a vision of mathematics towards those life sciences that goes beyond the classification from soft to hard. And this mathematical structure needs to take into account the complexity features of living systems, whether living systems are multicellular systems, immune competition, individual and collective learning, the modeling of crowds, of swarms, etc. And more importantly is that biological phenomena take place at different scales, which move from subcellular scale to the scale, which is the macroscopic scale at which the phenomenon is observed. So one cannot just take a macroscopic model that just reproduces the biological phenomenon without taking into account what's going on at the level of the cells, or even at in some cases at the subcellular scale. So when dealing with living system, really one needs to take into consideration all of those challenges, and in particular some of the complexity features that living systems have. The first one is the ability to express an activity. So the living particles, if you want, they're not living on their own. They communicate with each other, and they have feedback from the environment on them. And this ability to cooperate, to cope with the environment, is not the same for all the entities. So we always have some kind of heterogeneity in the system that we have. In addition, there is the nonlinearity of interactions, meaning that the interactions between the different parts of the system do not add linearly, and sometimes these interactions are non-local. So the effect on a certain particle can be seen even from something that is distant from that particle. Also, living systems have a learning ability, meaning this ability to cope with the environment is not fixed. It changes over time and evolves over time. So these are some of the main complexity features of living systems. And when one wants to model a living system, the modeling strategy needs to take into account all of these points. So once we start by the observation of the phenomenon from a macroscopic scale, we need to identify the different entities that are interacting, the type of interactions that is between them, to identify kind of the complexity features. And once we have these features, we need to decide what is the mathematical structure that is best fit to deal with such features. And then the next step would be to derive a model taking into account those features and the structure that we want to use, whether the structure is discrete, continuous, so it depends on the system that you have in hand. And finally, once we have the model, the next step would be the validation of the model. So the comparison of the model to quantitative comparison of the model to the data that is available or to the experiments that are available. So this is really in this introduction, I really wanted to lay out this rationale from which we will start with the modeling of bacterial communities. Well, so bacteria are, you probably all know, these are unicellular organisms, and they interact with each other to form really a complex system. So just to have an idea of the movies and the data that really led to our interest in the topic, and these movies were provided by a biologist at the Lebanese University, Qasim Hamzi, who approached us with these movies. So we see in this movie that we have an agar plate. At the center of the plate, we put an initial inoculum of some bacteria. Then the bacteria start growing. At some point, we see a fluid, a wave of fluid that emerges from the central place of the inoculum, and the density of the bacteria at the center indicates that bacteria are reproducing. We have the higher density of bacteria. Once this density reaches a certain threshold value, the fluid starts leaving from this region. There is an extraction, actually, a secretion of this fluid, which is known as surfactant. And then you have the buds that start emerging, and then we see those branching patterns of the bacteria. So what is the story behind all of this? Well, so the bacteria that we have is called Bacillus subtilis, but those branching patterns are not just unique to this type of bacteria. They are found in many organisms. So this is a picture of an agar plate on which bacteria was grown over a certain period of time, and we clearly see the fractal shapes that one can observe in non-living systems like in lightning or in snowflakes, etc. And this is a zoom to see how the dendrites they kind of avoid each other, and you have this very nice dendritic shape. So what is Bacillus subtilis? This is a rod-shaped bacterium of 0.7 micrometer in diameter to 1 to 2 micrometer in length, and it has a set of flagella along the cell so that it propels the particle in the medium. Well, and this set of flagella is not present in the initial inoculum of bacteria. Once the density of bacteria reaches a certain threshold value, the bacteria start differentiating, producing these flagella, and then they produce the liquid or the fluid, and then finally they start swimming to generate those nice branches. Well, several models exist. So this is what we care about, models that generate such patterns. And these models are not just restricted to those bacteria, but the goal behind studying such bacteria is eventually studying morphogenesis in multicellular organisms. So the existing model started as early as the 80s. After the extensive work of Matsushita and co-workers, which is a group of physicists in Japan who were interested in Bacillus subtilis because actually Bacillus subtilis is a bacterium also that is used for the production of enzymes, artificial enzymes, and for the production of a famous food in Japan, which is called natto. So that's why many of the references many of the studies go back to that group in Japan. So these bacteria show a cooperative behavior of self-self physical interaction. As we saw, we have the collective production of wetting fluid, and possibly some chemical signaling may be present and to form all of these different shapes. So in this morphology diagram that was constructed by Matsushita and his co-workers, we see that with a high agar concentration, so on the x-axis we have the inverse of the agar concentration, on the y-axis we have the nutrient concentration, the food that is available for bacteria. We see that under high agar concentration means that the medium is rigid, bacteria cannot easily move. So under high agar concentration, we see that these fractal shapes can originate. Under lower agar concentration, we see this is called a dense branching pattern. We really have very thin branches. Under high nutrient levels, but with high agar concentration, we have more like a compact growth with some kind of instabilities on the outer surface. Compact growth is observed under low agar concentration, medium nutrients concentration, and we have in some cases these concentric ring patterns showing some periodicity in the way how bacteria are moving. So this is the observation on one side. Now we move to the models. Again, the very first models were by Matsushita and Fujikawa, who were motivated to use atomistic models by the work on the zinc metal leaves that are obtained by electrodeposition. And in that case, they studied the zinc metal leaves in the context of diffusion limited aggregation, or GLA. And similarly, they studied the bacteria in the same context, and they showed that under low nutrient concentration, the fractal dimension of the bacterial colony that is grown is 1.72, which is consistent with the GLA structure. But what is the difference between, again, to just stress what is the difference between those GLA structures? So what is GLA? So you start with a seed in some place, and you have some particles that are diffusing, like in the case of electrodeposition of zinc. And these particles move and start aggregating on the initial seed to form these shapes, which are fractals. And at the time, it was the beginning of the computer simulations of those processes, the diffusion limited aggregation processes, and that's why they use this approach. Now, what is the difference between when bacteria are going and when we have a diffusion limited aggregation process? The point is that bacteria are growing from the inoculum, from the inside towards the outside. So we cannot think of the bacteria that is moving towards the seed. That's the first point of difference. The second point of difference is that these can only generate the fractal shapes which are observed under low nutrient concentration. They cannot generate the compact growth or the dense branching patterns. And the third point is, due to these experiments, they speculated that there must be a certain gradient that is involved in the generation of those patterns, and the gradient was attributed to the nutrient, the gradient of nutrient concentration. Now, another breakthrough in modeling bacterial communities was by Ben Jacob and his workers, who used the communicating workers method. So the communicating workers method was a hybridization between the continuous approach and the discrete approach. So we have the diffusion equation for the concentration of nutrients, and they represented the bacteria by walkers, but by what they call walkers, which is each walker is a bunch of bacteria that is moving at each step time a distance d with an angle theta, where theta is uniformly distributed over the interval 0 to pi. The bacteria have their internal degrees of freedom represented by this energy, which is the energy obtained from the consumption of food. And based on this model, they were able to generate these simulations. So all of these are results of simulations. Now, NC, because we have a walker that is supposed to be moving inside an envelope. So NC is, you have here at the outer sides, you have an envelope for this, for the inoculum, if you want, for the first representation of the inoculum. And then when the walker starts hitting the envelope, the NC is a threshold value beyond which, once the walker hits the envelope, a segment of the envelope, for it to move outside the envelope, you need to hit it NC times. So this is a threshold value that is really representing, again, the agar rigidity, or the concentration of agar in the medium. And they really succeeded in obtaining many branching patterns based on this model. But what was missing is that under low nutrient concentration, so P here stands for peptone, which is the nutrient used in agar for those bacteria. Under low nutrient concentration, they were not able to obtain some dense branching patterns that are observed in experiments. And to do that, they also introduced another term to the equation, which is related to chemotactic signaling. So again, chemotaxis means the movement of the bacteria or the cell towards a gradient of a chemical, either towards when this is chemoattraction, or away when this is chemorepulsion. So they introduced this in order to obtain the dense branching patterns. However, experimentally, it was not possible to detect the type of long-range chemotaxis that they proposed in their model. Now, another class of models is the macroscale description, the description from the perspective of reaction diffusion model. So since you have so many bacteria in the zone, if you want, we can represent bacteria by their density, which is U of xt here. And the change in this density is due to migration term plus birth minus death. So the movement is represented by a diffusion, which can be either linear or nonlinear. Birth is the bacterial reproduction, and death is the change of bacteria from an active state into a non-moving state or a passive state. So one of the first models that were used is the Fischer-Kolmogorov model. And here we have a linear diffusion term, and the birth and death are both summarized in this function f of u, which is u into 1 minus u. Now, in this case, with simulation, the branching was not obtained. We only obtain a compact growth in that model. So the next step was to introduce to this Fischer-Kolmogorov model the diffusion of nutrient, which was also known as the diffusive Fischer-Kolmogorov. And again, so in the case of introducing nutrients, we have another reaction diffusion equation for the nutrient density V, or nutrient concentration V, and eta is the conversion ratio of food into bacteria. So that's we have the change, we have the minus sign here. Now, again, even with this nutrient-based model, the growth was compact. There's no instability in the propagation of the front, of the diffusion front. And the speculation was that the model did not impose a minimal bacterial density in the colony that is needed to generate the fluid and then later on to generate the branching patterns. So to overcome this, Kessler and Levine proposed to introduce a cutoff function. So H is the Heaviside step function and beta is the threshold density for growth. Now, once this was introduced in the model, the simulations showed that there is some kind of instability appearing at the front that is appearing here, but this is not really the branching that we want to see. It's only some kind of what they called fjords. So the surface is broken by these cliffs, if you want, and it's not really a dancing, a branching pattern that is observed. So the next step was to introduce the death term in the model. So once they introduced the death term, meaning the change again of bacteria from an active state to a passive state to a frozen state, so these bacteria that are in the frozen or non-moving state are assumed to be non-moving. They're not moving, they're not diffusing, so they stay in their place and they do not reproduce. There's no reproduction term here. And once they introduced this death term, they really obtained some kind of branching patterns in the system and in the propagating front. Now, what is the reason really? The reason is that once they introduced those passive bacteria, these passive bacteria cannot diffuse from the center and cannot close the fjords that were introduced with the cutoff function. So this allows the branches to form. So this is one class of models that succeeded again in generating those branches. Now, another perspective was to use, without the cutoff function, to use different functions for the birth and death, like in, for example, the Gray-Scott model or Mimura's model. So in the Gray-Scott model, the death term is simply a constant, and we have this function for the birth that depends again on nutrient concentration. And in Mimura's model, we have the death term that kind of depends on it's limited by the high density of active bacteria or by the high density of nutrients. So in both of these models, when running again finite element simulations on these models, there was no need for the cutoff term to produce the branching pattern. So for example, in the set of simulations by Mroue and co-workers, which they used the mixed finite elements on some unstructured triangular grids. So with the Gray-Scott model, we identify here in the first row a front of active bacteria that is moving. So the first row shows the active bacteria that are moving forward on a sector showing part of a plate. And on this row, we have the total population. So this is both passive and active bacteria. And in this row, we see the gradient of nutrient concentration. So nutrient that is getting depleted here at the center of the plate. Because of the depletion, active bacteria tend to move towards the regions where there are more nutrients. And we see how the nutrients get depleted towards the center. So really this model again depends on the existence of a nutrient gradient and the limitation of nutrients, in addition to this distinction between active bacteria that move forward and passive ones that do not move at all. Similarly, also in Mimura's model, so it's the same, the dots represent the active bacteria, and this is the total population. We see similar branching patterns, again, with a different set of functions, F and G. Now, also another class of models that was suggested simply used nonlinear diffusion. Why nonlinear diffusion? Because bacteria are supposed to be performing some kind of random walk. So this can be at the macro scale understood as a nonlinear diffusion. And there is also the birth and the death terms in these models. And with the first choice of the diffusion coefficient, which is some constant d0 u power k, this is the kind of branching that was obtained by Kitsunezaki's model, and this is what was obtained with Kawasaki's model. So, so still again, with different, so the point is, with different models, each time you change the model, you can still obtain a branching pattern. Although the models are really different in their nature. So which model really explains the phenomenon? That's a natural question. Which one of them explains this phenomenon? And another question is about the parameters that are used in the model. So they are really tuned so that this branching pattern is exhibited without having necessarily a real physical meaning. Also still another class of models which are assumed under the unlimited availability of nutrient is due to chemoattraction and chemorepulsion, where in the diffusion equation, again for bacteria, there is a term for a chemoattractant, which is represented by the concentration of chemoattractant Ca and a chemorepellent Cr, along with the diffusion equation for the attractant and repellent. Again, in this context, one obtains the dendritic shapes that we see in practice, but we have a more, if you want, a shape that is more interwoven for the active bacteria that are at the front, and the dendrites tend to be thinner in that case. So that's one thing. Which of these models really is explaining the phenomenon? That's one point. Another point is that with several of these models, if you run the simulation with different mesh size, you get as you make the mesh finer with different mesh size, you really observe that you have different shapes, different dendritic shapes. One is not the same as the other. So this may suggest that the shapes that are produced may be due to a lack of stability in the numerical scheme, for example, and not really because the model is supposed to generate those patterns as they are. Now, another thing is that the comparison between those models and the experiments. So as we said earlier, we have the dependence of the dendritic patterns on the grid used for computing. We have different mathematical models leading to similar shapes. And one thing is that even in the absence of nutrient limitation, the dendritic shapes were observed, while many of the majority of the models relies on really the limitation in nutrient concentration for these dendritic shapes to be created. So whenever we have no nutrient limitation, and this was reported, there must be some other kind of physical or chemical effect that is piloting this migration of bacteria and causing these shapes to emerge. And one possibility is really the effect of the fluid, which is known as surfactant. Another possibility is probably the agar that is used in the culture, and which was not efficiently studied. And it was also shown that there's no substance generating a long-range chemotactic movement that is identified experimentally, as was suggested by different models. And one more important thing is that the different diffusion models assume that the bacteria in the stems and in the center of the inoculum become inactive, so they don't move. But it was observed experimentally that these bacteria are not by no means inactive, so they are able to perform some kind of random walk, and some of them may even be able to divide, but at a lower level than the active bacteria at the fronts. So there are different perspectives for the possibilities of what is the source of these branching patterns. And to be able to understand this, one possibility is to understand the motion of a single bacterium and how it interplays with the community. There is a need to study the structure of the agar surface and its wetting properties and its consequences on the local motility of bacteria. And similarly, there must be some models that take into account the different cell types that are available. So biologists started now comparing between the swarmers and the supporters that support, there is some kind of supporters that come from the inside of the colony and cause a certain drift that really helps moving the active bacteria towards the front. So there is a need really for a model that takes into account the several population density fields of bacteria. Now, by no means this means that the existing models are not that efficient. And to say this, I didn't find better than the words of Mroue, the writer of the Bible of mathematical biology. So in his introduction of the book, he's saying if the use of a model stimulates experiments, even if the model turns out later to be wrong, then the model has been successful. It led to something, it led to more experiments. And an important part of this statement is that the mathematics is dictated by biology and not vice versa. So there's always the need to look at what's going on first at the biological level in order to translate this into mathematics, not the opposite, impose the mathematics on the biology. Now, this is the context from which we started our review for the existing literature about the topic. And we started with our approach towards this topic in a slightly different way. So this is a work in progress. It's not a work that is done. And this is in collaboration with Adrien Dayer, who is a physicist from Université de Paris, Qasim Hamze, whom I mentioned his name already, he's a biologist from the Lebanese University, Ayman Murad, also from the Lebanese University. So we started from two different types of recordings that are available. Some are at the macroscopic scale that I showed earlier, and some are at the microscopic scale. So the strategy was to analyze the available data using image segmentation. So using image segmentation, we looked at the movies to extract the mask of the colony. And from this mask, one can find the area of the colony, the perimeter of the colony, and similarly find the number of branches that are emerging. So this is one movie that we already analyzed. This is another type that really shows the beauty of the shapes of the colonies that one can obtain. So again, we're extracting for every time frame, we are obtaining area, perimeter, and number of branches, how these are evolving in time. Now, the goal is to really obtain at some point the fractal dimension of these colonies. So in the gray movie, again, one can plot the area, how the change in area is obtained versus time, similarly perimeter versus time, similarly here for the blue movies, and this was done for different sets of movies. Also, we compared the evolution of area versus perimeter, and we saw a linear pattern in the relation between area versus perimeter. This is for the gray movie. For the blue movie, we had two different lines. So there is a change in slope from one linear pattern to another. For sure, we're facing many obstacles with the analysis of this data. Some of them are related to the fact that there is a change in zoom level in the movies that are generated. In other cases, there is the camera that suddenly moves away from part of the colony, so we don't have a full view of the colony. And that's why many of those experiments need to be repeated. So here I also show you how we are computing, how we are trying to isolate the number of branches going radially over the whole region and evolving in time by computing these intersection points between the red circles and the branches of the colony. And again, the goal that we have in mind is to use this data as a base later to compare the model that we propose to the available data and try to check the validity of the model. Now, moving to the model. So the approach was to move from the micro scale to the top, not in the other way around. So from the micro scale to the top, why? Let's look at, again, this movie of what's going on at the tip of a dendrite. So at the tip of a dendrite, we have a large number of bacteria that are moving. It seems like they're forming some whorls and they are at the same time reproducing. So there is the reproduction, the division of bacteria, they're both, their movement in groups like this. And the nice thing is, if you look at the boundary, you clearly see that whenever a bacterium reaches the boundary, it takes this kind of tangent position to the boundary, leaving what is inside not to leave this region. And as they are dividing and moving as such, we see how the front is moving down, how the growth is happening. So from a pedagogical point of view, if you want, or from the point of view of divide and conquer, the point was to start and try to reproduce something like that. So we wanted to start from the tip of the dendrite and try to reproduce this kind of motion in a step-by-step way. So we started by first as a group of bacteria, 2 or 3 bacteria that are moving in a certain region bounded by this boundary, which is simply an obstacle for us. And each bacterium has a preferred direction of movement, which is parallel to the longitudinal axis of the bacteria, and with small randomness. There is always, when 2 bacteria meet each other at some point, the bacterium waits until one of them moves in direction so that it's not in the path. So we have kind of bodies, rigid bodies that are moving around, and whenever they hitch it, they kind of come very close to each other. One of them stops, and then until the other moves away. And we are also simulating here the division of bacteria. So bacteria will grow and divide into half at some point, but we're not taking into account the motion of the fluid. So if we look at what we obtained in this simulation, so we're keeping track of the green bacteria just for the sake of visualization. So we have these that are moving around when they hit the boundary. So this is an artificial boundary that doesn't move. They simply go back towards the inside of the region. So we see they are growing, they are dividing. So even when, in this case, the number of bacteria becomes larger, we don't see really any rotational movement as what was observed in the movie. So the next step was the speculation that to introduce this rotational movie, we really need the effect of the motion of the fluid in this context. So again, even with a large number of bacteria, we don't see any rotational motion. So the next step, again, was to introduce the motion of the fluid, which was introduced by coupling that system of bacteria that are dividing to the Navier-Stokes equations, and we assume that the fluid is incompressible. Again, the bacteria were considered as rigid bodies subject to two kinds of forces. The first force is the self-motility force. The second force is the force of transportation that is applied by the flow that is caused by the fluid. And again, bacteria are allowed to grow by increasing their size to split into two equal shapes. And the motion of bacteria, the point is the motion of bacteria within the fluid is causing a certain set of forces, and this set of forces will influence the fluid flow. One bacterium moves almost parallel to its body orientation when there are no obstacles, and when there is an obstacle, it stops until the other bacterium moves away. And whenever they face each other, they simply move both of them in the opposite direction. And this is just to give you an idea of the equations that we are solving. So we have omega is the fluid domain where the bacteria are swimming. And at any instant time t, we have NB of t is the number of bacteria that we have. Xi is the position, Vi is the velocity, gamma is the acceleration, omega is the angular velocity of the ith bacterium. So we're dealing with every bacterium as an entity on its own. Fi is the self-motility force of the ith bacterium, Ri is the direction vector, and U and P are respectively the velocity and the pressure of the fluid that we have. So again, this is the Navier-Stokes equation, assuming that the fluid is incompressible, so divergence U is 0, and the forces exerted by the bacteria on the fluid appear in this term. Solving this system of equations at every time step, this is what we observe. So you clearly see as the bacteria is moving, these arrows indicate the velocity of the fluid in which they are moving. So for the boundary conditions, we're using non-slip boundary conditions, homogeneous boundary conditions on the boundary for the fluid. For the bacteria, we're using periodic boundary conditions. So when a bacterium leaves from one side, it enters from the other side. We clearly see that with this, we started, so due to this interaction between the motion of the fluid and the motion of the bacteria, we see that bacteria started at some point organizing into groups that are having some kind of rotational motion. So this is what we have reached so far in our model. As a next step, the next step will be to introduce the effect of the forces exerted by the bacteria and the motion of the fluid on the boundary. So this will be later done by moving the boundary due to these forces exerted by bacteria using the level set method in order to, and we expect that at some point we will obtain those fractal shapes of bacterial colonies. That's the first next step. And once we have this in hand, we can start comparing between the model and the experiments by comparing the fractal dimension for the experiments and for what we can obtain from the model. Once we validate our model, the next step is to try to obtain some kind of a continuous model by using the homogenization approach, which is kind of a coarse graining, where you have a limiting process by which you extract a continuous model from the discrete model that we have in hand. Once we have the continuous model, we need to analyze the effect of the different parameters on the model and to to analyze the stability of the system of differential equations that we expect to get in this model. So thank you for listening. And again, these are some references that were used throughout this presentation. And there are for sure more references that can be referred to in this context. Thank you for your attention.
Thank you, Fatima. You really gave a very beautiful illustration of our expectations for this colloquia, giving us a very nice and gentle overview of the subject, and then moving to the details of the calculations. This is really remarkable. We have some time for questions, please. Yes, sir, please.
Question on the last part of the simulation that you started when you started talking about your students to the your simulations were practically based on simple laws of interaction that has just stop and move, right?
Yeah.
We know that there are other ways to impose like local rules that could induce this rotation in the food. Like you could say impose alignment or like additional coordination rules
between the different cells, you mean?
I mean, it's an easier way than to copy it to the Navier cells. So additional interaction rules between the bacteria on top of what they stop and move. You can allow for alignment. You don't have repulsion as well, or attraction. Right? So these additional forces might help. And I think the work of Sweeney has some.
You mean when you have, this is probably in the context of multicellular organisms, when you have these attraction forces that help the different. The context of bacteria, they really have this random motion. I'm not aware how the possible adhesion forces may arise. Yeah.
It's not that they're moving and not caring about each other's motion. They somehow.
Yeah, this is possible, but yeah, this is possible for sure.
And for the last bit, when you want to compare your experiment with the model, do you have any other suggestion other than the fractal dimension as an identifier? How close your model is to the experiments?
We have area versus perimeter. We have possibly the analysis of in the model, if we introduce later more parameters like rigidity of, or we change the fluid, or we change the possible rigidity of the medium in case we introduce that. We can probably compare the branching patterns that are observed with the experiments.
There's the radial speed as well.
Yes, there is the velocity also of the propagation of the front and so on. Thank you.
Sarah,
Yes, thank you. Thank you, Fatima. Nice talk. So you have then two simulations you have done. So the first, can you go back to the first simulation, please?
Yes.
So this simulation is based on which model?
This is simply motion of bacteria. There is
So
only the motion.
You exposed before several models.
Yeah. No, this is simply a motion of bacteria. You're looking at entities, kind of individual based. Each one has its own properties. You have every bacterium is on its own, and it has this moving motion rules. It moves. It's propelled. It moves parallel to its axis, and with a small possibility of randomness, kind of a Gaussian distribution with a small. None of the
first one, which is like a kinetic model.
No.
So here, how do you simulate this? I mean, what kind of mathematics is behind?
Simply the equations of motion.
You have
motion for each one.
And each one will split?
Yes, each one is split
so
at some point.
The program must follow each
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 one.
one.
Yes, yeah. The nice thing about when you run such simulations, you have really you are numbering the bacteria, so you really know each one, where is it at which time step, and
Okay.
and so on.
So you track each one.
You can
So-
track everyone.
But the concept, we benefit from the matrix structure of MATLAB.
Okay, so you use MATLAB.
Yes, yes.
So here there is no finite elements.
No,
free
nothing,
form,
nothing.
All right, now, so then this is, and then you go to simulation 2, then you get the Navier-Stokes.
Yes,
So then you have to, you put another layer on that one.
yes,
That's what you have done?
yes.
So
You
have-
put another layer, which is you introduce the Navier-Stokes equations. And you have a grid here. We're using simply finite differences with a splitting
So you
method.
use what?
Finite differences.
Finite differences?
Yes.
Okay, so what, on this?
On a square region.
On a square. Okay.
Yes, on a
This is
rectangular
this is
region.
very boundary condition.
Yes.
Okay.
Periodic for the bacteria. For the Navier-Stokes, we're using a non-slip boundary condition.
So now, what are the perspectives? What do you want
So-
to do?
so the
For example, all
perspective-
models you have, excuse me,
Yeah.
all the models you have discussed previously, what will you do with that?
So the models is just a review of the existing literature. So the strategy is really completely different. You're starting from microscale and you're looking for kind of, after you have the microscale model, that you try to analyze that it's really reproducing what you expect to observe. You try to get a continuous model by homogenization, meaning by passing to the limit at some point. And then once you have this homogenized, which is a macroscopic model, you can compare it to the existing models. You can compare, well, what is the diffusion coefficient in my case, if there is, and which kind. So we expect some kind of stochastic equations to arise, not just deterministic equations.
Why do you care about the continuous models? If, I mean, the way that I understood you were just simulating them. Why don't you want to just have a discrete model?
I do not understand what you're. Yes, please.
So why do you need to have a continuous model?
Why do I need? Well, usually when you have a continuous model, it's you have the different possibilities of analyzing the model, the stability, and so on. So it will better provide probably an understanding of the model and what's going on. The discrete works very well in generating the features probably, but to understand the behavior of the model, it's always kind of preferable. So if you want to study stability and so on, it's usually passing to the continuous case.
Simple enough that you can analyze their stability.
Which ones?
Let's say the original.
The original continuous model. Yes, they are simple. You can analyze their stability, but the problem is that it's not understood really. They tune the parameters to generate, for example, a certain branching pattern. But what is the effect? It's a phenomenological description. So we usually compare between phenomenological, physiological. So it's a phenomenological description of what you are seeing. So when you change a certain parameter, what does this mean? So for a physicist or for a biologist working on the experiment, it's really important to, when you want to compare your model to his experiments, to understand, well, what is the effect of that parameter? Why is it doing that? So you need this relation between the two, and usually we prefer not to have phenomenological models. Similarly, we had the same experience with cardiac electrophysiology. When you use a phenomenological model, you can reproduce so many things, but the point is, if you have a disease, if you have a certain illness that is causing a certain cell, for example, not to take calcium enough, not to produce some ions enough, and so on. So what is the, not to use efficiently some ions and so on. So how do you see this in your phenomenological model? There isn't really, you cannot make this connection. So that's really the problem when you're dealing with biology.
Given those continuous models, how can you be sure that the simulations that you showed are really the thing that the equation doesn't, that those simulations are not? Like what you said, how do you know that? Because those simulations are
So how do you know that in your model, in the proposed continuous model that you can try to reach, you're not also doing the same thing as the existing models? That's your point?
How do you know that continuous model corresponds to
reality? Because when you start, yeah, the point is when you start from the bottom scale up, you know already the parameters that you are using, what is their meaning?
Yeah, so
okay.
Your approach, I agree.
Yeah.
The original one. I mean, the older ones that you were reviewing at the beginning. How do they know that corresponds to?
Well, they try, for example, to study, for example, the stability. And they expect, for example, that this function has a different fixed point, for example, that are not stable points for the system. And then you expect that under some conditions the stability will be lost. So you have a propagating front that is unstable. That's how they started. Yani they started if you want, imposing the functions that will generate this by studying really the behavior of the function or the different critical points of the functions and so on.
If I may, I think they're just mostly motivated by the observation or maybe the assumption or the belief that similar behavior must be underlined by similar mathematics. So they go with this, then the reality tells them otherwise, which is what Mroue was trying to indicate in this statement. I think mostly this is what's going on. But in this case, I think they're trying to do a bit more diligent about it. Whether it works out or not remains to be seen. But
yes.
Initiative. Any other questions here?
Okay, so as I understood, one of the motivations to introduce the Navier-Stokes equation was to also see these tangential areas that they.
No, not yet really. It's to see the rotational movement.
Right. So as of now, the model doesn't also explain these tangential.
No. Even the biologists really do not understand why are these standing. So what is the reason that you have these bacteria that stand as such and kind of inhibit the other ones from moving forward, even from a biological point of view? Possible, but I have no explanation at my own level.
And so, what exactly was using Navier-Stokes, the motivation to use that, was that just for rotation?
No, and to see also the effect, because there is a fluid that is secreted at some point. So you saw in the movie that there is a certain fluid. So you need to see the effect. How is this fluid helping the motion? And this is not just observed, I think, in the case of bacteria. For example, one can speculate that whenever you have a flock of birds, why do they move all together? So, yeah.
So my question is, are there any systems that behave the same? So is there a universality class that spans the motion of the bacteria and other systems?
Well, you can think of, crowds at the level of bigger organisms. I don't think that they do this rotational movement anyway, but this is a motion in crowd. You can think about it as motion in crowd, but you don't have these.
For example, the model and the transition between gas and liquid and gas and solid.
Yes,
so
there is in phase transition.
Understanding of what we can span the information from here to information from there. Can we do the same with this system?
So the point is that they started in this direction. So when they drew the morphology diagram, it was really kind of like the phase diagram, if you want, that is observed in that case. But really the complexity of living systems is different. So you cannot approach it just as in the case of as inert matter. So that's probably the difficulty.
Would it be fair to say that diffusion limited aggregation is the some sense, the simplest general mathematical realization of display.
Under limitation of nutrients.
Yeah, we forget about reality now.
Yeah.
But when it comes to mathematical models that display such dendritic behavior.
That's for crystals. That's for inert matter. Yes.
Yeah, that's probably the context in which physicists want to start in. But there is another context actually, which is the Safnan-Taylor stability. You're familiar with it?
Not yet.
Yeah. So this is the stability which involves a front between different fluids, and surface tension can drive instabilities that produce beautiful dendritic behavior.
Like Marangoni forces, you mean, or something like that?
They can be associated with that, sure, because they bring about variability in the tension, and they bring about dendritic instabilities, fingerings of this nature, etc., etc. Now, for instance, did you try to wonder whether bacteria have any surface tension of their own?
That's being studied actually by Adrian. What is normally they have experiments where if you take a droplet of water on a window that is like this, you expect the droplet of water to stay sessile in its position. It happened that in some experiments, when you have the agar plate tilted with 1 degree, something as small as 1 degree, really the motion of the initial inoculum is really outside all expectations from the physicists.
In 2 plates?
No, on a plate when you're growing, and the plate is a bit tilted with even 1 degree. And even when you have a fluid that is standing like this on the glass, you expect the drop to stay in its position due to surface tension, etc. But so this is being under study. And
okay. So then one last question from my end on the mathematics of your model. So you have the bacteria on a torus, but the fluid on a square. How do you do that?
Bacteria on a torus.
You chose your boundary conditions for your bacteria.
Yeah.
And you have your fluid on a square.
Yeah.
What is the motivation for that?
Well, because because we still do not have
bacteria, you're not asking of your fluid. That introduces
but because we haven't introduced because we haven't introduced yet the boundary
so you have to be careful what sort of aberrations this is introducing.
Because we haven't introduced the boundary yet. So that's the point. Well, the next step is to introduce the boundary and how the boundary will move due to the motion. So in that case, you change the boundary conditions.
This is something you have to look into.
Yeah.
And then about the Navier-Stokes, why the full Navier-Stokes? Why not just the Stokes regime? And this is a highly damped situation. Bacteria are moving with very, very low Reynolds number. Why are you solving full Navier-Stokes?
I have no answer.
You can do it, right? It's cheap to do it.
Yeah.
I think one can learn a lot by looking at Stokes regime. There's probably.
One last question. So for the video where the pump is moving, this vortex. But you stop at the size of this vortex is the real video. No. You mean the microscopic video, the video at the tip? No. No, not yet.
Not there.
No,
there is a lot to be done. The point is there is a lot to be done.
I'm glad to see all of this excitement. Thank you very much, Fatima. This was a wonderful excursion.
Thank you.
All the best with your project, and we look forward to the workshop in the fall. Just to say that the series of colloquia by CAS fellows will continue. We'll have one last lecture for this session with Sarah Najm talking about network complexity. This is what, in a couple of weeks?
Next week.
June 8th? June 8th, so June 8th. So we look forward to that. And in the meantime, we have a visitor of CMAX who is going to give us a couple of lectures on topics related. Yes, this is Leo. So this is coming up next week. And we're very much looking forward to your talks. It builds on collaboration between CMAC and Leo, and builds also on the overview lecture that CMAC gave us a couple of weeks ago. So this is all very exciting. Thank you very, very much for your participation, and enjoy the end of your semester.
Thank you.
Thank you for